Good afternoon, everyone. I think I will just start. What a truly remarkable scientific feast this has been. Thank you, thank you. And I don't know about you, but I truly immersed in the brilliance of our speakers, and I hardly, noticed time passing, and now we're at the final session of today's program, our prize award ceremony and winner presentations. So this moment brings me back to early 2023, when ChatGPT really got the attention from the world. And that, reminds us and also feed us with a great idea, an idea that what if we create a Nobel Prize like AI Prize for science? So we approached to our longtime partner, Science Magazine, and shared the idea. And I'm very glad that the editorial team embraced the idea, and Yuri, stepped forward generously to lead this effort. And this is how this prize was born. So, and one thing I want to tell about is the prize each year. Some people ask me the question. Each year, the prize honors a grand prize winner and 2 finalists whose creativity and vision represents the very best of AI accelerated research in their fields. And from the beginning, we know if you want to create a prize from scratch, the right partner is important, so critical to the success. You have to work with someone with deep scientific credibility, but also have a global platform powerful enough to elevate emerging voices, support early career scientists. That is why our collaboration with science has been so meaningful. Together, we have built a prize and a platform that amplifies the impact of early career researchers who are shaping the future of science. And now it's my great pleasure to introduce Dr. Teresa Maldonado, President of AAAS, to the stage and say a few words. Thank you.
Chrissy, thank you so much. I'm very excited about this prize. First of all, the vision by the Chens to create this foundation and to support this work in such a generous way to improve the human experience by understanding our brain. I mean, that's just huge. The partnership with AAAS, the American Association for the Advancement of Science, as Chrissy mentioned, I think is a powerful way to launch the importance of this prize and the visibility. For those of you who are early career, I'd like to point out that I have been a member of AAAS for over 40 years, and I encourage you to join as students. You'll have access to the Science Journal, which will open your aperture to amazing stories of science based on rigorous evidence. I'm also excited about this prize because it recognizes early career researchers. You are our future. And in this era of AI, where discovery and results are accelerating at such a pace never seen before, we need to work together across the generations, but we need the fresh lens of our new researchers, and I'm so excited for our finalists tonight. I'm really eager to hear from you. The rising stars are our future. And to look at AI in accelerating science, absolutely, since that has true meaning to society. Society is eager to learn and benefit from our work, and it's our responsibility to deliver it in the most responsible way. So this prize encapsulates the values that I just summarized. So with that, I'd like to move forward and introduce, well, I won't be introducing, but our 3 recipients for the prize today. And with that, I'm going to ask Yuri Solomonov to the stage. Thank you.
Hi everyone, my name is Yuri Suleimanov. I'm a senior editor at Science Magazine, and I had the privilege of serving as a managing editor of this prize this year, managing reviewing process. So I guess I can use my time in your time, to tell you some ideas about applications that we received. So it's a new prize, and despite that, we received quite numerous applications from different countries around the world. So almost half of them was from United States, but we did see quite a few submissions from China, Canada, United Kingdom, Australia, Singapore, India, countries of European Union such as Spain, Italy, France, Germany, Netherlands, Belgium. And also because the unique feature of this prize, it has no constraints on scientific disciplines as long as research is quiet? I'm too quiet? Oh, okay, I'm sorry. I'm sorry about that. So what I was saying, that this was very international set of applications that we received. And another beauty of this prize is that it is not constrained to a specific scientific discipline as long as we're talking about research that was AI-driven, enabled, or accelerated. So we received quite a diverse set of topics, essays with quite a diverse set of topics. So for instance, we had many essays from healthcare, telehealth, nursing, clinical decisions, disease diagnosis such as sclerosis, brain tumor, early dementia, allergy, rare diseases, mental health care, psychopathology, neurology, heart development, medical imaging, phenotyping, material science and chemistry, inorganic materials, complex molecules, autonomous laboratories, biogeochemistry, agroecology, computer science, AI foundation models, biology, molecular network biology, neuroscience, behavioral studies, neurodynamics, cognition neuroscience, psychology, physics, quantum computing. I should have done that. I'm sorry. Quantum computing, astrophysics, social sciences, human communication, large language models, and social sciences, and even civil engineering. So it was quite a diverse set of essays that we received. I guess by showing this, I wanted to say that it's obvious that machine learning is entering different fields. But also I wanted, by showing this, to thank the people who were responsible for promoting and advertising this new prize in different scientific communities. I think they did a great job. And also to thank all the applicants who submitted their essays. You know, writing essays, I think, is way more challenging than preparing a research paper, because we used to write research papers, but essay is a different thing. And I really enjoyed reading quite an interesting set of essays from different topics. Many of them were of very high quality, but nevertheless, we had a very thorough review process, which involved consultations with my colleagues from Science Magazine, but also with external experts. And I guess I need to emphasize here that the whole reviewing process was completely independent from the Chen Institute. And at the end of this reviewing process, we had a clear picture of our top 3, 2 finalists and the winner, and I'm very happy that we can meet here today in person and congratulate them on winning these awards and to enjoy their talks about their exciting research. And with that said, I would like to ask to come back to the stage Chrissy and Therese. Please welcome AI Prize finalist Alicia Robert. Please welcome another AI Prize finalist, Aditya Nair. And finally, please welcome the grand prize winner, Joran Kiao. I'll present. All right, now we can proceed with the presentations from our winners, and we'll start with Alizée Robert, a researcher from the Flanders Merwin Institute. She uses machine learning to map how coastal water absorbs carbon dioxide, while oceans are known to absorb about 1/4 of human CO2 emissions. Coastal regions remain poorly understood. Alizée combines satellite and in situ data with AI to generate high-quality detailed CO2 maps, 10 times more accurate than previous models. And her work helps clarify coastal water's role in carbon cycle and supports better environmental decision-making. Right now, Alizée is refining the methods with high-resolution maps and data to further enhance our understanding of carbon exchange in oceans.
Okay, thank you very much. Before starting my presentation, I would just like to mention that I'm very honored to receive this prize, and I would like to thank the CHEN Institute for the recognition of my work and to show how we can use AI in marine science. Thank you very much again. So now I'm inviting all of you to dive into the ocean, and I will show you how can we use AI, how we can take advantage of AI in marine science. So I like to start my presentation with this nice picture of a planet taken it from the moon. Because with this picture, we can really see that the ocean, our blue planet, the ocean, is a major component of a planet. The ocean plays an important role in many aspects of a society and for a planet. But today, I will show you one of the most important roles of the ocean, from my personal opinion, is that the ocean help us to regulate the climate through the carbon cycle climate feedback. So today we know that the ocean is absorbing a large quantity of carbon dioxide, CO2, which is one of the major greenhouse gases in the atmosphere. In a quantitative term, the ocean is absorbing every year around 1/4 of the human-made carbon dioxide which is emitted to the atmosphere from fossil fuel combustion and land use change. So in another term, the ocean plays an important ecosystem service to the human society because the ocean helps us to regulate the climate. So in our field, we say it through the marine carbon cycle and the climate feedbacks. But the question now is that how can we quantify this number, 1/4? How can we have this global number? How do we know that in total the ocean is absorbing 1/4 of human-made CO2? How can we approach this exchange of CO2 from the atmosphere and the surface of the ocean? Are all of the regions in the ocean absorbing the same quantity of CO2? Are there regions who emit CO2 to the atmosphere? What about the temporal variability of this exchange, from the seasonal time scale to the interannual to decadal time scale? How this absorption of CO2 change over time, and how in the future this absorption will change over time due to climate change, but also human perturbation. And what about the coastal seas? So the coastal seas are close to the land, so you have also an influence of the land through the river discharge. The complexity of the coastal ocean is still less and poorly understood compared to the open ocean. How can we address this question, but based only on observation? So not using like a mechanistic model. How can we quantify globally the contribution of the coastal sea or the open ocean to the carbon cycle climate feedbacks? So to tackle this question, we first need measurement. So here on this slide, you can see different possibilities, different techniques to measure the concentration of CO2 in the surface of the ocean. And if you have the concentration in the surface of the ocean and the atmosphere, we can calculate this exchange of CO2. So there are different techniques. The first one is to implement instruments on research vessels. So here you have a nice animation of the Siemens Steven, which is the research vessel from the Vlies Institute, the Institute for Maritime Research. We can also have some instruments on moorings, on a ship of opportunity, who perform the same transect in the ocean, and then you can add instrument on it and measure CO2 in the water. You can also ask to the citizen to make a collaboration with scientists, like with a sail drone, and then we can add instrument on this sail drone, which is a famous race, Le Vendée des Globes. It's a super famous sail drone race that crossed the Southern Ocean. And then by adding instrument on the sail drone, we can collect data in the ocean. So here, on this slide, you can see an animation of all of these transects that has been done in the ocean through this research vessel and ship of opportunities. So you have transects from the west coast of the US, the east coast of the US, you have transects that cross the Atlantic Ocean. So it's like a monthly maps from 1950 to today when you have all of these transects. And since 2002, we also have these floats that collect also an important measure of carbon dioxide, temperature, salinity. So each dot that you can see in the ocean is one of these floats. And they just follow the currents, they take measurement, and then they give back this measurement through satellite observation. So you can see that we have some region when we have a lot of data, a lot of transects from the ship of opportunities, the research vessel, but also some region like in the Indian Ocean when we don't have so many data. So all of these data are collected, this global initiative. When we collect all of this data of CO2 in the surface of the ocean, we synthesize and we quality control, and then we collect it in a huge database called the Surface Ocean CO2 database, the SOCAT database. So what you can see here is all of the measurements that we collect in the ocean from 1970 to today. The color value represents just the absolute value of the CO2 concentration. So you can see that in some region we have like the red colors, that mean that the concentration of CO2 in the ocean is quite high, while we have in the blue region, the concentration of the CO2 is quite low in the ocean. So you have like an important spatial heterogeneity of the concentration of CO2. But the most important thing is that you can see in these figures that some regions are quite well covered in space, in terms of observation, while we have other regions, like in the Indian Ocean or in the Southern Pacific, when we have like some missing data, some region when we have zero collection of data. But if you look again in this database, which has really monthly maps, every month they synthesize all of the measurements. If we split into the different years, you can see that this database is in continuous in space, but also in time. So if you look at the measurement taken in the 1970, you have very poor data compared to the early years when you have an increased number of data collected in the ocean. So this SOCAT database is in continuous in time and space. So if you have an order of magnitude, the SOCAT database covers only 2% of the surface of the ocean in total over the long period since the beginning of the SOCAT. So it's discontinuous monthly maps of all of the observation that we collect in the ocean in terms of CO2 concentration. So how can we approach at the global scale how much the ocean absorbs CO2, emits CO2, this temporal variability, based only on this incontinuous CO2 measurements. So you can have like a lot of uncertainties because this database is very incontinuous in time and space. And then this is how we can use machine learning approach. So with the machine learning approach, we can fill the gaps in time and space, and we can provide monthly maps of the CO2 concentration in the ocean, everywhere in the ocean, every region is covered, and every month of the year. So how does that work? So the machine learning approach that we use take the advantage of this database, the SOCAT database, all of these in situ measurements that we are taking in the ocean, combined with satellite observation. So satellite observation of other variables of the ocean, like sea surface temperature, salinity of the ocean, chlorophyll A of the ocean, because we know that the concentration of CO2, which differ from one region to another one, we can have a low concentration in high latitude, high concentration in along the equator. We know that the concentration, the value of the concentration of CO2 depends on the physical chemical and biological properties in the ocean. Biology will take up CO2 through photosynthesis. The temperature will affect the PCO2 value. In low temperature region, the PCO2 is lower compared to high temperature region because of the solubility of this gas in the water. So we will take the advantage of the spatially and temporally resolved satellites record from several variables that we know that they drive CO2 in the ocean. And then through this advantage of in situ measurement and satellite data, we're going to construct a bridge through a machine learning approach, the self-organizing map and the feed-forward neural network, to construct complex, nonlinear relationship between this proxy, this variable that we know that they control the concentration of CO2 in the ocean. And then when this relationship are reconstructed from different regions through the SUM approach, self-organizing map, we are able to apply this relationship in region when we have no value of the concentration of CO2 in the ocean. And then through this machine learning approach, we are able to reconstruct spatial and temporally resolved maps of the concentration of the CO2 in the ocean, and here more specifically for the coastal seas. So we have this spatial and temporally resolved concentration of CO2 in the ocean, in the coastal ocean. If we measure the concentration of CO2 in the atmosphere through other methods, we are able to quantify this flux, this exchange of CO2 from the atmosphere on the surface of the ocean. And this is what we have done and what is shown in this animation. So here what you can see, it's monthly maps from 1982 to today of this exchange of CO2 between the atmosphere and the surface of the ocean, specifically designed for the coastal sea. So in red, you can see that this shallow sea or coastal ocean that emits CO2 to the atmosphere, and then in blue is the coastal region would take up CO2 from the atmosphere. So through AI, through this AI bridge, we are able to reconstruct like high spatial and temporal resolved flux map of CO2 from between the coastal ocean and the atmosphere. And with this, we're able to quantify the global contribution of the coastal ocean to the marine carbon cycle climate feedbacks, the temporal evolution of this coastal carbon cycle, and how we can identify how the coastal ocean responds over time from one region to another one. So for instance, you can see like we have the Amazon, we have like this blue plume, which is like moving from one month to another one, is due to the salinity change in the Amazon plume, which will affect the exchange of CO2 between the atmosphere and the ocean. We also have along the African coast, we have this very intense red area. This is the upwelling region, when you have like CO2 from the deep water who come up to the surface and emit CO2 to the atmosphere. There's a nice noise on the left. Give a bit of flow. So to wrap up, to conclude, with this example, we show that combining in situ measurement, satellite observation, and this bridge that I construct through AI, we are able to better quantify, I would say, the health of our coastal ocean, as an example, through the CO2 analysis. And then these kind of methods can also be applied to other variables which are very important to track, like for instance the pH, the acidification of the ocean is also important to track how acids become our coastal ocean in the future. We can also track the oxygen, because oxygen concentration in the ocean is very important for the marine development. So I think this AI approach really opened the door to also resolve and better constrain the health of a coastal ocean. And here in this example, I only discuss about the surface of the ocean, how the surface of the ocean exchange CO2 with the atmosphere. But I think we can also in the future also go through depth and with the AI approach to resolve how the CO2 can then, below the surface, can be exchanged with the water depth. So this is the conclusion, the wrap-up of my presentation. And I just would like to finish with also this quote from Wunsch et al. 2013 that even if we have the development of all of this AI approach, I think it's very important to mention that observations not taken today are lost forever, because the basis of this reconstruction is the measurement at sea. So it's important to continue to go at sea, to take measurements, to improve the different tools that we use to take measurements, to improve the temporal resolution, to deploy instruments everywhere in the ocean, because without observation, I think my AI bridge can also employ a lot of uncertainties. So I would like to thank two institutions, because this work has been done with the University of Libre de Bruxelles and also my actual institution, which is the Flanders Marine Institute, the VLIS. And again, thank you very much for the recognition of this work and this prize. I'm very grateful. Thank you.
Thank you, Alizée, and my congratulations again. Questions? We have some time, please.
Hi. Nice presentation. I was wondering if your method, the method that you use for self-organizing my map and feed-forward, can be used for land measurement of CO2 or other atmospheric components. A direct application I think I have in mind, because I work for an agricultural company, is the model, the yield. If CO2 is yield of crops, like, for example, CO2 could be a limiting factor or directly affecting yield. This will improve a lot of a predictive model, I think, in general.
Yeah.
Have you thought about or know anything that directly use the similar method for land chemical estimate?
So I didn't really compare our methods to the land, because I think for the land it's quite more complex, because we also have like CO2 emission from the human activity. We mixed, with the natural, I would say, variability of carbon dioxide. But I think it's a good strategy to think in the future, indeed.
So it's indeed true that the land situation is much more complex than the ocean.
I think, yes, it's more complex.
Thank you.
More questions? No?
Yeah, thank you for the presentation. That answer just inspired this question, which is, so oceans also have human-produced CO2 sources, if far fewer than lands, like shipping or other economic activity. Is this added complexity like a source of error you have to deal with or take special concern with, or is it just like sort of small enough to be in the background noise? Like, how does this factor into your modeling decisions and that sort of thing?
So if I understand your question, is that how can I separate the human contribution to the natural variability? And from the ocean perspective, we measure the total CO2 exchange. So we are not able to split between human and natural variability. And this is why I think we are our field of expertise is relevant with the model development, the mechanistic model development, because with our reconstruction, we can better constrain the mechanistic model, and then with this model, we can split between the anthropogenic contribution and the natural contribution of the marine carbon cycle, I would say.
Mm-hmm.
So it's a complementary approach.
But do we know how much is...
Human contribution isn't too big an issue to cause you like much modeling problems?
Say again?
So the human contribution in ocean is like not large enough to cause you too much modeling problems that you have to deal with it separately or anything like that?
Yeah.
All right, thanks.
Thank you. All right, we can switch to next speaker. Thank you, Alizée, again. Just need to say a few words about next speaker. So our next speaker is AI prize finalist Adiya Nair, and he uses machine learning to uncover hidden patterns in brain activity related to emotions like anger and fear. While individual neurons show little correlation to emotional states, This model analyzes collective neuroactivity, like listening to an orchestra, to detect persistent emotional signals. His work revealed the so-called line attractor pattern in hypothalamus, offering new ways to quantify emotional intensity over time. This breakthrough could improve psychiatric drug development and expand how researchers interpret brain data. IDEAS is also building tools to help scientists apply AI models more easily, making advanced neuroanalysis accessible across the research community. Adin, please.
All right. So firstly, thank you again so much for this opportunity. It's such an honor to be standing among so many of my scientific heroes, and I'm just so grateful that I have this opportunity. So today I'm going to take you on a journey of how we've combined neuroscience with AI to unravel the neural basis of emotion. Think about the last time you felt angry, the last time you felt afraid. These experiences are really complex. They're filled with feelings, they're filled with different types of experiences. And yet, hidden within that complexity are certain common qualities that are present across emotion states. We call these qualities as emotion primitives, and they allow us to study emotions even in animals like mice. These qualities include things like escalation, which allows an emotion state to have a graded level of intensity, as well as persistence, which is why if you become angry, you tend to stay that way for often several minutes. These qualities are often at the heart of many mental health disorders, and so we've been searching for where and how these are being created in the brain for a long time. A lot of this work has honed in on a structure which is deep in the brain in a region called the hypothalamus. And what we've uncovered are that small groups of neurons in subregions like the ventromedial hypothalamus, or VMHPL, play an outsized role in the controls of emotion like aggression. We know this from lesion studies and efforts to turn on and off these regions. And so to really figure out what these neurons are doing and how they orchestrate emotions like aggression, in the lab we perform our experiments where we can monitor the activity of hundreds of neurons as the animals are naturally engaging in behaviors like anger and aggressive behaviors. And to make sense of this, we can look at the activity and compare that to the behaviors that the animals perform. So in this plot over here, the white lines show you the time-evolving activity of different neurons, and these are using calcium imaging, a different form of signaling. And the red lines here show you when the animal performs aggressive behaviors towards the other animal in the cage. And to our surprise, we couldn't really find a clear correlation between the animal's activity and the behaviors that they perform. In fact, across animals, very tiny fractions of neurons were robustly encoding or signaling when the animal was performing its aggressive behaviors. This was a real paradox to us because we knew that this region was important for anger and aggression, and yet at the level of individual neurons, it was really difficult to find these signals. And we wondered whether new tools in AI could look into this type of dense population activity and reveal these signals. What we want to do here is discover the governing equations that explain the dynamics of activity. But we want to do this in a physics-inspired, informed way, as well as using interpretable ML techniques that allow us to actually make predictions and perform perturbations to causally validate whether these models represent true models of neural circuits. And to do this, we use a different class of state space models that approximate nonlinear dynamics as a set of interacting linear dynamical systems. The way these systems interact are through nonlinear switching rules, with our recent approaches in this space using Gaussian processes to actually regulate state switching. But they give us a handle now to fully understand the system because the underlying states are still linear. So let's see how this works at the level of our activity. So we start with our high-dimensional activity, which often can be hundreds of neurons. But we fit our models to a reduced-dimensional neural state space. Here we identify different patterns of activity that share certain common properties. These are like melodies of an orchestra, where different instruments come together to create certain patterns and certain symphonies. But our models are attempting to discover why these trajectories in the brain are moving in this particular way. When we discover this, we can recover what we call the flow field or the vector field. These arrows show you how activity in the brain is predicted to move in this space in the absence of any input. And they showed us, in this case, across animals, that activity was always converging to a set of stable points in the system. These stable points are called attractors. They're ways of encoding persistent variables in the brain, as well as in neural networks. And because they're arranged like this in a line, this type of system is what we call a line attractor. Another way of visualizing this is as an energy landscape, where we take those arrows and we project them into a 3D space. And this reveals the shape of a trough. And we saw this in every animal, and as activity traversed along this trough, the animals displayed progressive aggressive behavior. So to make sense of these models, we can rely on the fact that these models are fully interpretable. We can take those linear states and really identify exactly the neural patterns that correlate with the line attractor. And this reveals to us the neural signal that look like that of an aggressive state. It was initially really low, and it ramped up over 100 seconds as animals were escalating their aggressive states. But it peaks when the animal performs aggressive behaviors like attack towards the other animal. But notice that this activity also stays stable during these interbout intervals. And so here's a signal in the brain of the animal that stays high even when the animal is not expressing overt behaviors like aggression. And we think this is actually tracking an escalating and persistent aggressive state. And now this gives us a handle to study these type of internal states in mice for the very first time. These type of models are nice, but they're correlational. They're data-driven discoveries, and they're ultimately hypotheses. And we need to prove that these models are truly true models of neural circuits. And it's important because models of attractors have been influential in early AI, work by John Hopfield and Sebastian Sum, among others, but there's been very little evidence that the brain actually uses attractor dynamics, in particular line attractor dynamics. To actually show this, we've had to innovate and create a closed-loop platform where we can record activity, model that activity using our dynamical models, and then try to play that activity back using a system called holographic optogenetics. This allows us to effectively play a piano in the brain, where we can activate individual neurons, which are informed by our model in this case. So now we can record that same activity that I showed you before. This is that same slowly escalating signal that I showed you, now in a different mouse. In this case, that signal also has a slow de-escalation phase, representing that de-escalation of an emotion state. But now we can try to recreate that. And by using the predictions of our model to activate exactly the right group of neurons, we can start recreating this. In this plot over here, each of these white lines show you when I activate a certain subset of neurons that are predicted by the model to have this feature. And you can see that with every repeated activation, the activity goes up, but it stays high because neurons are actually interacting within the subcircuit. And with every subsequent activation, it keeps going higher and higher. And so you can recreate this quality of persistence. And when you stop that activation, there's this intrinsic decay of activity, just like we see in natural dynamics. And so by showing these properties, we can validate key predictions of line attractor dynamics, and in fact, obtain some of the very first causal evidence that the brain, in fact, truly uses a line attractor to encode different signals. We can also show that the way that these cells interact were actually dependent on the release of slow neuropeptides. By merging our ability to record and model activity with CRISPR to knock off receptors for neuropeptides, like oxytocin and vasopressin, what we could show are that these receptors, these slow neuropeptides, were important for maintaining the persistence of our attractor dynamics. And so now in a series of studies, we've gone from data-driven discovery, causal evidence that these are being used in the brain, and even aspects of mechanistic dissection. And what's exciting to us is that these dynamics don't seem to be specific to anger or aggression. In separate studies, we've shown that these dynamics are also present when encoding sexual arousal in the brains of female mice. And by recording these dynamics through the estrous cycle, we see some dramatic transformations in these signals in a way that actually correlates with the estrous cycle. And when taken together with our studies in aggression, as well as some studies that we're doing in fear, we think that line attractors could be a general principle that the brain is reusing to encode both the escalation and persistence of different emotion states. Now, what's particularly exciting for us is that for many decades now, psychiatrists have long hypothesized that impairments in attractor dynamics could underlie different mental health disorders. For instance, many psychologists have hypothesized that conditions like depression and anxiety could actually be a result of plasticity of attractor dynamics. You can think about an energy landscape like this in the brain, which becomes deeper when when you have an impaired state like depression. And as a result, activity is persistently present in that deeper attractor well, and it's really difficult for activity to escape. And now we can discover these changes in a data-driven way, and we're excited to start embarking on that in more recent work. But to do this, we need tools which can be employed at scale, not just in our lab, but also labs across the world. And to do this, we're creating new agentic AI systems, which allow researchers across the world to be able to apply these methods in a much more user-friendly manner, using fine-tuned large language models to perform an end-to-end analysis of activity data using natural language. We're also creating tools that allow users to do this for behavior data. All neuroscientists have locked up in some hard disk somewhere hundreds of hours of video data that they're yet to analyze. And to solve these issues, we're creating new transformer-based architecture models that can basically reveal different types of affective behavior straight from data. What's been exciting about these methods are that as we dissect these models, we've been uncovering recently that these vision transformers that have been trained to recognize behavior also start looking a lot like the brain. In fact, their intermediate layers possess representations that look exactly like the line attractor, suggesting that certain deep models could even be foundation models to predict activity in the brain and eventually be used in models like digital twins. And so that's our hope for the future, that these studies that look at basic computations in the brain underlying affect could lead to the development of new foundation models and bring us into this new domain of computational neuropsychiatry. And so with that, I have many important people to thank. This type of work only happens in the intersection of different fields, and that's something I really got to do in my PhD in the lab of David J. Anderson, guided by many computational mentors, including Scott Lindemann at Stanford, Pietro Perona at Caltech, as well as Ann Kennedy currently at Scripps. I also want to highlight some phenomenal neuroscientists I got to work with, who I collaborated with really closely, including Amit Vinograd, Mangyu Liu, and George Mutafaris. I just started my lab now in Singapore at the Lee Kong Chian School of Medicine at NTU Singapore, and we're continuing a lot of this work, especially in the development of foundation models. So if you're interested in, if you have any questions, I'd be happy to answer that.
Thank you so much, Adi, and my congratulations again. Questions?
So, first of all, thank you very much for the presentation. It was incredibly interesting, especially the part where you programmed the brains of mice to feel rage. I had a question about that specific intervention. So it seems to me like you find sort of linear vectors in mouse brain embedding space, where when you add those vectors to the brains of the live mouse backwards, you increase the aggressiveness score that you're recording off of their brains. So my first question is, do the mice then become behaviorally aggressive?
That's a good question. So behavioral causality of that is something we're still exploring. It's hard because when we do these experiments, the mice are head fixed, and so there's literally a microscope on its head, and they can't overtly perform behaviors towards other mice. There are ways of circumventing this that we're looking into, but that's an important question that we're trying to pursue right now.
Yeah, all right. And then my second question is, have you looked into investigating the degree of transferability between mouse brains of those linear factors?
Yeah, that's a really good question. So we fit these models in an animal by animal basis, but one of the most fascinating things we see are that even in genetically inbred mice, different mice display aggression in different ways. Some mice, when they become angry, they tend to stay angry for large amounts of time, just like us. And other mice, when they become angry, that decay of an aggressive state is really fast. And so in the models that we fit, we find correlates to this. For example, the attractors of mice that are extremely angry and stay angry for large amounts of time, those attractors are very stable. The energy landscape is particularly flat. Versus in animals where the aggression decays really quickly, the attractor landscape is a lot more tilted. It's a lot more leaky in a sense. And so there are these correlates to individual differences in aggression that we're finding in attractor dynamics, and it's an exciting direction for us too.
Yeah, so one really interesting and relevant discovery in natural language models and computational models has been that models can be stitched together with affine transformations. So you take first half of one model, second half of the other model, and you learn an affine transformation on a few data points, and then it works to stitch them on other data points. So that would be really interesting if that held true for mouse brains as well.
That's something we're looking into, and especially how do you combine activity across animals, but there's also this degree of flexibility you need to build in because behaviors are different across different mice. And so those affine transforms need to actually take into account some form of flexibility, and we're looking into that and having hyperparameters that tweak, let's say, one parameter of an attractor state across animals.
All right, thank you very much.
Thank you.
So if I could just ask one very brief question. When you stimulate the brain and observe the persistence of that effect, have you looked at controlling that for, say, stimulating other areas of the brain where you would not expect persistence and observing whether or not it still persists just because you shocked the brain?
Yeah, so it's a great question. So specificity of these circuits is really important. So if we actually record from this structure, we see these persistent signals. If we record from other structures, we don't see the same. And so we know that both from the stimulation results, like you alluded to, but also from these imaging or recording experiments. So it's not that every part of the brain has a persistent signal that regulates an affect-like state, but different neural circuits are using different codes. And in fact, other regions that are still related to aggression are actually encoding other aspects of aggression, like the motor component. And so other regions are actually reading out these type of persistent signals and converting them into the motor action. And so there's different codes in different parts.
Thank you.
I think she was first. So.
Hi, thank you so much. That was really interesting. I'm a child and adolescent psychiatrist and a neuroscientist, and so, of course, my bias is just in thinking about how our psychiatric diseases develop. Typically, they develop very early, and we see kind of early changes in development. Have you guys started to look at kind of how these programs develop in the brain and kind of where they go awry?
Yeah, so that's a really interesting question. I mean, there's a certain interesting machine learning component to this, which is that a lot of times when you think about these computations, you think of them as truly learned in the experience of the organism's lifetime. And yet a lot of these systems we're studying are truly innate. They're pre-programmed. And so we're thinking about how is it that these neural networks are actually pre-programmed through a genomic bottleneck? And how is it that maybe certain differences in those miswiring could actually result in impairments at the level of, for example, adolescence and so on. So that's something we're thinking about a lot, and it's also forcing us to go in interesting directions in ML, because it's all about how do you feed in some kind of inductive bias into a model that allows them to have these features. So we're thinking about that a lot.
Awesome, thank you.
Hi, thank you for the presentation. I have a question follow-up with the specificity. He mentioned areas. I want to mention different cell types. I see you bring up TCAM6, which is, I'm assuming you're using in vivo calcium imaging as a main method. So is that introduced by genomic editing or introduced by virus? So follow up with that, do you plan to look into different cell types, especially interneurons, action in your system? And how would you fit your AI models with different cell types, including interneurons? And I came up with another question is, estrogen, and I think in couple slides you mentioned gender differences with the emotions. We know estrogen influence them, and we also know they influence memories too. I worked with Gary Lynch in UC Irvine before, so so I have more interest in how do you explain those memory models into AI if possible. Thank you.
These are all interesting questions. I'll answer them really quickly. So for the first part, we use viruses to introduce these different genetically indicated encoded calcium indicators. We do this in a cell type specific manner. So of all the studies I've described so far, we're targeting estrogen 1 receptor expressing neurons in this particular region in the hypothalamus. What do other cell types do is a really important question. In the subcortex, things work a little differently from the cortex. In the cortex, there's this very canonical structure where you have glutamatergic neurons and you have different classes of interneurons. And the hypothalamus is a little bit more difficult to describe what is an interneuron, which is what's a true projection neuron, because those markers are not really a one-to-one match. But we've been looking into other regions, other cell types that express, for example, inhibitory markers that surround this region, and we've been looking into that. But that is an interesting question. This last question about what estrogen does, I mean, one of the interesting aspects of this work is how we're starting to tie hormonal signaling to computations in the brain, because those type of signaling have usually been thought of to work at a much more slower time scale, and people don't think of that as computation in the same way that people think about fast computations like memory, right? So that's something we're starting to see, and in our studies in female brains where we look at changes across the estrous cycle, these attractor states are strongly modulated. So for example, in states where the estrogen levels are low, the attractor is actually lost. So there is an important dependence of the attractor state on hormonal signaling, and we're looking into the mechanistic basis of that now.
All right, thank you.
All right, let's thank the speaker again, and my congratulations, Adi. All right, so our final speaker today is AI Prize Grant winner, Zhorang Qiao, and he uses generative AI to model dynamic protein structures and their interactions with small molecules, dramatically accelerating drug discovery. Inspired by diffusion models, his approach predicts not only protein folding, but also binding behaviors, enabling the design of new treatments, including undruggable diseases. His work replaces years of experimental effort with seconds of computation, creating a computational microscope to visualize molecular systems. Now, leading chair discovery, Qiao is a building tool that automate drug design and explore novel therapeutic pathways, transforming how scientists understand, stimulate, and create molecules in a post-AlphaFold era of digital biology. Zhorang, please.
Good afternoon. Thank you, Yuri, for the kind introduction. First of all, it's my great honor to be awarded the Chen Institute and Science AI Prize for 2025, to be sitting with all of the greatest figures of science right here. And it's my great pleasure to have the opportunity to share my research about building better AI models to build a foundation layer for predicting molecular structures and reprogram some of the biology at the molecular level. And I also would like to take this as an opportunity to share some of my reflections on where it fits into the greater evolving picture of AI for biological science, or the so-called tech bio field. So if you look a bit back in time and see how the field of AI biology has grown so far, we are truly living in an incredible time of unlocking new capabilities for both fundamental science and the industry. So we are almost having an entire tool stack to be able to digitize the entire central dogma and having tools spanning across genomic sequences to their structure and functions of the proteins, and they're just continuing to become better. If you're from a research background of an adjacent field, you probably have been seeing many technical talks in this field. And here for the 10 minutes I'm granted, I'd like to share more about my perspectives on how we got here and what motivated my own research. So find the beginning. So what made all of this entire technological stack possible is that structural biology has become computable. What structure prediction is providing for science is really like a microscope that can help us decode the hidden, obscure language of biological sequences that were picked up through evolution and transform them into three-dimensional shapes formed by atoms, which is what human scientists are trained to understand and rearrange based on what we learned as the language of physics and chemistry. And the reason predicting protein structure from AI was possible is that there's an underlying energetic principle behind natural evolution, namely the residues that are staying close to each other in the folded structure also has to mutate together. So this really defines the biological sequences we nowadays observe from all of the living species. So we are left with this daunting learning problem of mapping these massive noisy evolutionary traces back to the protein folds, which is actually a perfect fit for deep learning algorithms. So since the release of AlphaFold 2, the leading research figures of this field are quickly enabled to brute force a large fraction of all of the known proteome and greatly speed up how we engineer new proteins like Professor Dave Baker introduced earlier today. But solving protein folding is not solving molecular biology. Proteins don't carry out their functions in isolation, and they form assemblies and ordered compartments across multiple scales. What this means is we need to expand the capabilities of the computational microscope we are just talking about to go beyond single structures and actually mapping out those linkages between the individual proteins, which often drives a phenomenon at a higher level. For example, for those interactome protein interactions, some problematic linkages can drive the cell into a pathogenic state, and we have to understand their interactions between proteins at atomistic resolution in order to understand how to reprogram those, how to find agents in order to put those into and reverse them into a normal cell. And in terms of how they respond to such chemical perturbations, proteins are dynamic molecules that change conformations by binding to other species, including small molecules, nucleic acids, and post-translational modifications. Some of the well-studied examples for those conformational changing proteins include ATP synthase and GPCRs that actually cycle through multiple conformations in order to transduce energies and stimuli. From Jennifer Doudna's talk, we also learned that CRISPR is also a beautiful example in this class. So we'll need to represent general molecules beyond those proteins that are made up of just 20 amino acids. And we should not just be predicting a single answer because that is not how it actually functions in normal cells. So this complexity has been a challenge for many traditional computational strategies like molecular docking, which often assume that the binding occurs through a lock and key mechanism. Although molecular dynamics can model some binding and conformational changes at the same time, it is also a problem to overcome these slow kinetic barriers between the low-lying conformational states. So being trained as a computational chemist, I was actually fortunate to learn about many of the mitigations to this problem developed since the 1990s. And many of those working techniques involves running simulation over kind of like a gradient of temperatures in parallel to better decorrelate those samples, and as well as like guiding the sampling with prior knowledge about the system, for example, those experimental data like NMR shifts or those co-evolution signals. So think about this, we are actually having a philosophy for modeling those biomolecular systems kind of similar to how we are doing text-to-image generation. Nowadays, we are doing a conditional diffusion model for biomolecular structure sampling. So starting from here, we want to bring a solution to these problems, and we aim to develop a unified model to predict biomolecular structures by folding all of the species of these biomolecules of interest all at once into a distribution of outputs, and we can efficiently sample hypotheses from this distribution and cover the statistical ensemble beyond the frozen snapshot. Several key innovations we put into this model are essentially just inspired by what we were just talking, those biophysical principles and breakthroughs in generative modeling. And part of that is being able to condition on multimodal inputs, covering those evolutionary traces, language models, and a careful embedding of the atoms, bonds, and molecular chirality. And to read out the structures from those conditioning signals, instead of using a kind of a handwritten set of regression rules, like in previous folding networks, we built a chemically universal diffusion module to sample these atomic coordinates of the complex, regardless of whether the atom is coming from one type of molecule, protein or small molecule or the other. And finally, we engineered the neural network in a way to make it agnostic to the physical transformations, the symmetry transformations you apply on the system, namely translations and rotations. In other words, like we built this system to be equivariant. And part of this encoding network is to be able to model the pairwise interactions and communicate those pairwise interactions to each other in order to refine the hypothesis about the binding interface. So we introduced this iterative mechanism so the model can learn to gradually mix information flows from different length scales together and continue to infer the long-range interactions among different species. The resulting model we obtained in early 2023, we evaluated an existing problem for protein ligand binding that the field used for testing out those molecular docking softwares. And we are quickly seeing this universal generative diffusion approach to show strong promise. And encouragingly, Neuroplexer, the first version, reached better accuracy than almost all of the other methods we tried at the time. Of course, we know this is going to be the first model we developed rather than the last one, and it will be the worst one. It will have many caveats. But we ask ourselves, if we prompt it with some existing knowledge, can we already extract some insights? So one of the examples the model performed well is KRAS G12C, which is a covalent inhibitor. So there's a covalent inhibitor opening up a binding pocket that will be invisible from the static structure alone. And as we continue to improve the model, we also found them to show near experimental agreement with over half of the GPCR structures it was never trained on if we prompted with a proper ligand. Later on, my team working with many great scientists and engineers at Alembic decided to switch to a more scalable flow matching architecture and investigating efforts and compute to establish the so-called compute optimal scaling laws across a mixture of data modalities. Namely, those are almost all of the pairwise combinations of the type of interface different molecules can make with each other. We realized that if we really be careful about the data mixture and improve the data quality while perform like a careful post-processing, these models were actually very far from saturating its performance. And as a result of these heavy lifting scaling studies, we reached a state-of-the-art model that almost excelled across the full spectrum of biological structures. Evaluating it against AlphaFold 3, which were launched earlier 2024, the same year, the results were highly competitive, especially for drug-ligand co-folding prediction and for covalent modifications, which actually matters for many detailed biological processes involving those like, for example, metal cofactors. It's also worth noting that confidence estimation has become an important part of structure prediction folding workflows people use nowadays. As what Professor Anima highlighted earlier this morning, confidence estimation and being able to provide multiple hypotheses with a proper confidence score is really important for human scientists to interpret the result and make decisions on top of those. What we and many groups found out is that these confidence measurements in the context of folding models are not only useful for interpreting the data, they're often also predictive of experimental measurements of interactions like those from binding affinity, even without additional training. This has been opening up many opportunities for designing novel ligands. Fundamentally, what the models have picked up are statistics. They can be incredibly accurate and useful while still missing some aspects that are obvious to a chemical physicist. So some examples are like protonation, salvation effects, and chemical reaction, for example, proton transfers, that are often associated with bond breaking. Fortunately, the tools are being developed. For example, we have built some geometric deep learning models for electronic structure, which can accurately predict the potential energy surface of complex molecules involving bond breaking based on fast approximations. We can also apply some simple physics even at the model prediction stage to guide prediction at an inference time in order to make it more accurate. As we productionize the system, pushing speed and efficiency was just as important as the technical capabilities. To showcase an example of how we can address special optimization challenges for molecular structure predictions, where all of the pairwise interactions and their representation needs to attend to each other, our team created a fused triangular attention module with smart tiling and recomputation techniques to dramatically reduce memory and compute usage. This makes it possible to handle larger structures and faster predictions. Finally, Structure Foundation model has learned about protein evolution and interactions from existing molecules. How do we actually accelerate the way we design new molecules? And how do we actually deliver on the promise of using AI to help drug discovery? I think a lot of these questions eventually boil down to whether we can bring generative capabilities to these foundation models and actually being able to reason about functions and create new bioactive molecules from scratch. So we are really proud of the progress we have brought to this field, to this binder design problem. And one of the greatest leap of performance this year is the zero-shot antibody design models released by the Chai team, including myself, where we achieve a double-digit binding success rate in the lab when generating antibody binders for antigens without any known antibodies, which is an over 100-fold improvement of success rate over previous state-of-the-art. The success rate of Chai 2 means we can nowadays experimentally validate binders with just a small-scale experiment custom-made for a novel target within a scope of just 2 weeks, which is a big step over traditional antibody engineering techniques like yeast display or animal immunization, which takes millions of dollars and multiple months of lab work to perform the discovery. So to conclude my talk, speaking of the future, what are the next frontiers to work on? First of all, the dynamic problem and statistical ensemble prediction problem is very far from being exactly solved. Now, given that we know what are the chemical compositions of a bioassembly, we're having reasonably accurate tools to predict their structures. But what if we don't know what molecules are in there? We have to go back and think about what are the biological mechanisms driving the formation of those complexes. I'll be also eager to see extending these capabilities to more sophisticated interactions and arbitrary stoichiometry. Some of the examples include those protein-protein interaction interfaces stabilized by chemical modulators or induced proximity. And solving many of those problems can in turn help us reprogram protein interaction networks, the interactome, and design ligands that can modulate the subcellular, for example, localization of pathogenic proteins. From a therapeutic point of view, this approach to rewiring cellular signaling may also hold some promise for better therapeutic window by selectively targeting the certain states of proteins while sparing those that are essential for normal cellular functions. So at the end, I would like to thank again for the Chan Institute and Science for making all of this great conference happening, and say thanks to my mentors from Caltech, Tom Miller and Animan Kumar, who are both on the audience seat, and many of my former colleagues from Iambic, collaborators from NVIDIA, and my current colleagues from Chai. Happy to take any questions.
Thank you so much, Joran, and my congratulations again. Great work. Questions? Don't be shy. Okay, I guess the talk was excellent. It was everybody understood everything. I guess now we can conclude the session. I would like Jennifer to come to the stage. Yeah.
Can I ask a quick question?
Oh, I'm sorry, I overlooked. Oh, Tom,
Yeah,
please.
I just had a
Tom.
question.
Hi, Tom.
So, based on your experience, can you talk about the relative promise of generative AI for small molecule design versus large molecule design? Can you speak to the relative advantages of those modalities and the way that they are more or less amenable to a generative AI approach?
Yeah. I think I would really like to call the year of 2025 a year for generative protein design. And part of the reason of that is like, we are having pretty powerful architectures in order to model the conformational, structural, and sequence distributions for biological sequences. And this relied on development of like being able to train on two sources of data. One is those structures from PDB, and also those sequences from, for example, UniProt, all of those like genomic sequencing data. And extending that to small molecules would require us to also like broaden the scope of the models and improving its capabilities to more sophisticated chemistry beyond the code book of amino acids. And the second problem we need to solve is chemical synthesis. So for protein production nowadays, you can go through like mammalian expression, cloning, and go through those binding assays in a relatively, I would say, commoditized manner. But for small molecules, you also have to think about what is the right conditions, the right synthetic route in order to actually create those molecules in the lab. So I can't be more excited about actually solving that problem. I think someone like on the audience seat may be having some great ideas about actually like reaching the next milestone in small molecule drug design.
No more questions? Sure. All right. Thank you so much again, and my congratulations on yes.